Okay, so today I'm going to be going through a presentation that I will be doing tomorrow for Journal Club. It's my first ever real journal club, so I'm a little nervous about it, but so I'm here to practice it. Okay, so the paper that I'm looking at is named Xist RNP drives sex-biased autoimmunity in mice and humans. Here we have a little outline of all the things that we're going to go through. So we're going to go through the introduction and then each of the figures in the paper, and in total there's 7 figures, so we're going to be here for a minute, but background. So X chromosome silencing is a thing in mammalian females, and it's where in humans, it's randomly chosen which X chromosome is going to get silenced, but it gets silenced to make sure that the gene expression is pretty much the same between males and females for the sex chromosomes. So it's like a dosage compensation thing, so you don't want too much expression of the X chromosome genes, and so your body silences it, and the main driver for the silencing is this Xist gene, or long non-coding RNA. So this is a gene that is expressed only in the inactivated X chromosome, and it forms sort of this huge chromatin cover around the inactive X chromosome, forming this bar body that you might have heard of before. So here's another representation of an X chromosome, and so here we have an active X chromosome. You can see there's a lot of genes that are available to express, so you can imagine that we're inside the nucleus and we're looking at the chromosome. Here we have this active X, and it's easy to grab onto things and transcribe, where here we have this bar body where there's only a few things that's able to be accessed, right? So one of these things will definitely be the Xist, but there are other genes that sort of escape this silencing, and so those are shown in other colors. Okay, so we know about X chromosome silencing. Xist is something that is specific to females. We also, the reason why we care, or why the authors are looking into this, is because there's a huge sex disparity between males and females for a lot of different diseases. So in autoimmune diseases specifically, you have a huge bias for females getting it and having more chronic illness compared to males. So one of the most cited sort of diseases is the systematic lupus erythematosus, or SLE. It has a 9-to-1 ratio, 9 female to 1 male, and I think in some cases, for more specific lupus types, it can even get up to 11-to-1. So there's a big sex disparity in autoimmune disease, and it seems like having this extra X chromosome is correlated to your susceptibility of disease. So obviously females have a bias, but also triple X syndrome females who have 3 X chromosomes, and then also Klinefelter syndrome males that have 2 X's and 1 Y chromosome, they are more likely to get autoimmune disease than people with sort of the normal amount of chromosomes. And so the authors hypothesized that this Xist RNA protein complex, or RNP, which is associated with that silencing, which is only present in females, is immunogenic and may underlie this female-based autoimmunity. Here is a little background on different proteins or components that are associated with this Xist RNP. So I think some of these are part of the RNP, but others are just associated with it. And so you can see for a lot of these, they're autoantigens, sorry. And so they're associated with different types of disease. So SLE is the one that is most, I guess, has the most of them. I don't know what this other is, but obviously there's a trend here where things that associate with the Xist RNP are also seen in autoimmune patients as autoantigens or as markers for this type of disease. So let's see if I got everything. Okay, yeah. Okay, so introduction. This is their sort of graphical abstract. So they're hypothesizing that in female cells, you have this cell death, and then all the components of the cell, like Xist RNP, are floating around in your body, and then T cells and B cells recognize this as something to sort of like attack or that are foreign, so they make antibodies against it, and that affects this autoimmune response, which targets their self. So that's pretty cool. The way that they decided to go about this was to look at mice. So we'll move on to the first figure here. It's kind of a lot to take in, so we're going to go one at a time. So they have the long non-coding RNA sequence of Xist, and to make it so the point is they want to somehow put this Xist long non-coding RNA in males to see the effect of it on like autoimmune disease. So but they don't want to have this silencing effect because it's going to like mess with a lot of your genome and your chromosome stuff. So they deleted this A repeated section, which is essential for like inactivation of a chromosome, so that like the coding for the bar body, that will happen even if you put this sequence on a autosome, so it will still silence it, which is interesting in itself. So they deleted this and they saw that it did not create this code, but it didn't affect the RNP. So they have this delta or mutant repeated A mutant Xist. So they took this sequence and they added this specific promoter. So the TET-OP is a promoter that is only activated when the what is it? When give me a second, sorry. Oh, doxycycline. So you here they refer to it as dox, so I will in the future. I have it right on the page. Yeah, so this promoter is specific to dox, and so it will only be expressed if there's dox in the system. And then they selected for heterozygous mutants. So since Xist is only expressed on one of the X chromosomes in wild type, they wanted to mimic the same sort of effect. So they chose a system where only one of the chromosomes has this Xist. So they do this, and then they see that these mice are, one, living, and two, have the phenotype that they're looking for. So they tested to make sure that the actual expression of the Xist was there in these male mice, and that this was similar to wild type males. And then and they tested specifically chromosome 11 expression. So they looked at wild type males chromosome 11 expression and then compared that to these mutant males, and they saw that it was about the same. And they also compared that to females with the this mutant also introduced to them. So they didn't add dox or anything like that. They're just looking at the baseline, and they saw that there wasn't any differences. And so they then went backwards and put this sort of they did this process in specific mice strains. So they used, well, we'll get into it. So that's what this figure A is explaining. Figure B is, okay, so they're looking to see if this mutant male actually expresses Xist and when you add dox, and to see if that's different from the mutant male when you don't add dox and the mutant female when you don't add dox. So ideally, orange and green should be about the same, and blue should be low, like very low. So here, they don't give error bar or like significance, but you can see the general trend that the females and the mutant males with dox have similar expression levels. So that's pretty nice. That's what we wanted to see. That's the goal, at least. And here it's pretty hard to see, but it's showing that we can see Xist with a marker. So instead of just measuring with, I think this was a qPCR, they also looked at FISH or fluorescent in situ hybridization. So they they were basically saying that this fluorescence of Xist in females was pretty similar to the expression of Xist in mutant males when you add docs. So, cool. We have a good setup. We have this mutant strain. We know that it's expressing Xist. We know that where it's inserted is not affecting the chromosome that it's inserted at, and so we have a good baseline. That's basically what this figure is saying. And can I move my head? I think I can move my head after the fact, but okay. So figure D is looking at Xist expression in different organs. Honestly, this is really hard to see because I don't know what is behind my head. 1 second. So, this is what I was explaining before, where they looked to see if there was Xist expression in these mutant males compared to wild type males. And so you see in all of the organs that they tested that the mutant males had this higher expression of Xist. I do think it's pretty interesting to see the disparity between each organ. Like liver doesn't seem like that is very significant, but I'm not sure. But in the spleen, it's like huge, the difference. So, and this was after 2 weeks of this. Sorry, this is from here. This is why we do a practice run beforehand. Okay, and then the last part of this figure, this part is by far the hardest to get through this in figure 2. After that, it should be pretty easy to comprehend. So here in E, they're just showing the setup of how they harvested the organs. So they treat the mice with docs, and pristane is a drug that induces like autoimmune disease. So yeah, so it just makes them more susceptible to autoimmune disease. So they collected blood, which then they'll do antibody profiling, and then collected the organs, and they did sequencing. So they did a TAC-seq, which is to identify chromatin states, and then RNA sequence to identify what's being expressed. And then in F, we have the actual cohorts. So we have this positive control of mutant females with no docs and this pristane addition. So in this case, we should see a pretty big phenotype. And then male wild type, so, and no docs, no pristine, just like straight wild type males. And then we have the mutant males with the pristine, but without the docs, and so uninduced mutant. And then we have wild type with the pristine added, and then we have the actual test where we have the mutant male, we add docs, we add pristine. So ideally, we would see that this test mouse has similar phenotypes to the positive control. Okay, figure 2. This is going through the sequencing that we mentioned earlier, so let's go through each of the figures individually. Here, A is just to sort of organize your thoughts, so it's just the different cohorts. And so B is MA plot, which is log ratio and mean average. That doesn't really mean anything. The biggest thing here is that it's comparing differences between groups. So it's a pretty common thing to use in genomics when you're looking at differential expression. And so MA plots and then also PCA plots are usually used to show this sort of if there's any disparities between groups. So red here, the red dots mean that there is a difference. And so it doesn't really matter if you're doing male over female or female over male. It should still, like the relative difference should stay the same. And so we see that in the test, the mutant male with dox is pretty similar to the mutant female without dox. And this is different than if you're just comparing males and females, and if you're comparing mutant males with dox to normal males. And then here we have a PCA plot which is explaining the variance in the data. So you can think of, if you're familiar with regression, like say linear regressions, you pull out different like beta values or like groups, variables that explain the difference between like variance in the data. So PCA or PC1 of 55 variance is pretty high. PCA or PC2 of 14 is, I would say, pretty average. But I think because of this plot, like PC1 is definitely the more sort of causal variable to describe this variance. And so you can think of the x-axis as this sort of range or spectrum of female phenotype to male phenotype. And I put this here based off of what we're seeing. So all the red are the female, and then the green are the tests. The blue are pristine mutant males without dox. Black is just wild type males. And purple is wild type males plus the pristine. And so it's pretty striking to see this very clean sort of spread between the groups. I'm sure they were really happy when they found this. And they're saying that these three mice were most like the female phenotype. Again, I think it matters more based off of the x-axes, but you do see this sort of clustering of females up here. So overall takeaway, the mutant males with dox are pretty similar to the wild type females, or the mutant females without dox. And here, I think I just copied the figure from figure 1F here to like show you the different groups, just to make things easier to understand. So here you see these color bars, and red is your positive control or your female mutant with pristine, and then green is your test, which is mutant male with dox and pristine, and then your uninduced mutant, your treatment control, and your negative control. And the point that we're supposed to take away here is that in terms of chromatin and expression, the positive control and the test seem to line up well. And here, so when you're reading heat plots, people usually like to like group things. So you can kind of see like, oh, these groups kind of go together and these groups don't, that kind of thing. So here we can see that in cluster 1, we have downregulation of some genes in the positive control. And we see that trend also in our test, but not in all of them, right? So, and then in our sort of negative controls, we see this overexpression of genes. So overexpression compared to low expression seems like these guys relate to each other a bit more. And then in cluster 2, it's the opposite. So certain genes are overexpressed in the positive control in our test, but downregulated in our negative controls. So they decided to pull out this cluster 2, which shows these sort of interesting groups, overexpression genes. And I forget what pipeline they used, but they did some type of gene ontology or pathway analysis, and they found that most of these genes that are overexpressed in positive and our test, positive control and our test group, were related to immune function. So that's kind of cool. It gives like sort of confidence that they're looking in the right direction, right? If you're seeing these sort of differences and they're related to immune function, they're probably hitting on something important. And same sort of takeaway here with the RNA sequence. So D is showing that there are similarities in chromatin state, and then F is showing the actual sequence. One thing to note here is that they don't have many mice in each cohort. So within the positive control, there's only 4 females, and one of the females is like pretty different than the other females, and so you can get a lot of variation even in your cohort. And then here G is, I forget what they used, let's see. Okay, so they based this fraction off of the RNA sequence that they got. So they predicted the frequencies of CD4 positive T cells, and most notable is that positive control does not have any of these. You have to look again because my head's there. It doesn't have any of the CD4 T native cells, and that this trend is also seen in the our test group. There are still some that have it, but so the frequencies of each of these subtypes are similar between our positive control and our test. And okay, so this is just a summary of figure 2. We're using CD4 positive T cells to get our attack seq and our RNA seq, and that's how we can do all of those different plots, whether it be the MA or PCA or the heat maps. And I just said that the chromatin is pretty similar between the Xist expressing males and females, and gene expression is also similar. And the TLR pathway, which I didn't highlight, but it was probably in that sort of list of gene pathways that they pulled out from their attack-seq data. And then I also mentioned this, so the frequencies of the different T cell subgroups were similar between the Xist expressing males and females. Okay, figure 3. We have a good setup of everything. We know our experimental setup, and now we can really start to see what the phenotypes are like. So we've established a general trend that these test males, these mutant males with dox and pristine, are similar to mutant females with pristine. So both of those groups are Xist expressing groups, and so we see this trend that in gene expression and in chromatin states, they're similar. Now we're going to go test actual disease phenotypes to see if they're similar as well. So here we have a different sort of setup. We're using mice that are more susceptible to autoimmune disease. So before we were using the C57BL6J mice, and here we're using SJLJ mice. So these mice are more prone to autoimmune disease. They chose 16 weeks as the cutoff for tests because they know that the sort of life expectancy of females with induced SLE is about 16 weeks. So they didn't want some of their samples dying and then losing sample size. So they chose that as the cutoff. And yeah, again, they did blood collection and organ harvesting. Same setup, just different mice strains and timelines. And here we have our mouse cohorts. Sorry, my legs are falling asleep. Okay, so our mouse cohorts are pretty different. Again, our positive control is not different. It's still, oh, it is different. Before it was female mutants without dox or pristine. Here it's just female wild type with pristine added. So I guess they didn't introduce the non-silencing version of Xist in chromosome 11 here. So it's just normal female mice. And then male, so we have a negative control, we're not doing anything to that guy. And then we have this dox control, so we have the mutant males and we're adding dox to induce the expression of Xist, but we're not adding pristine. So we're not making them sort of worse off. And then treatment control. Sorry, give me 1 second. Okay, so the treatment control is male wild type, so non-mutant with the doxin pristine, and then our test is the mutant with doxin pristine. Okay, so after these 16 weeks of treatment, they did sort of pathology scoring based off of histologies, I think. And so they counted up sort of damage for each of the group's organs, and so they gave each organ group this damage scale number, and they added them all up. And here you can see, oh, this is better. Here you can see that the difference between the females and the test male group was pretty similar. It's still significant, but much less significant than the other groups. So, and you can just look here, right? Like 9.38 is closer to 15.54 than any of the other groups, right? But it's kind of hard to see that, so they included some pictures of organ histology pictures. So the little dots or the arrows indicate something that's related to disease or the pathology. Honestly, I think it's pretty difficult to read these things, but if you're just comparing like visuals, this definitely looks more like this than the other ones, and I would say this also looks more similar to this, and that's just based off of like, you know, the number of holes or how defined this little circle is. And so they chose a cutoff of pathology score of 10 to say if something was severe or not. And so they chose these mutant males, our test group, as severe. And to note, like, not all of the males had this like severe pathology score, and so they did in the future separate these males, these test group males, into severe and non-severe. Yeah, so it looks like the trend is continuing, but it's also clear that these test group is not as sort of, it's not hitting the same level as these wild type females. Here, again, just to remind you what we're looking at, we have the cohorts. Here we're looking at the autoantibodies after 16 weeks. And so these autoantibodies are sort of known things that they're looking for SLE, like disease states. And again, you see this trend where, at least for all of them, you get this non-significant difference between the females and the positive control, which is pretty cool to see that, where there are differences between the positive, like the test, and the other males in the study. So this is again supporting their claim that at least the test group is making similar autoantibodies as the wild type female, and that these are happening at a different level than the other males in the study. So let me just make sure that I got everything I wanted to. Yeah, okay, so figure 3 summary, basically saying that, figure 3 is basically saying that after triggering this disease in these autoimmune immune deficient mice, right? Autoimmune prone mice. After you do this trigger with, which is the pristine or pristane, you see that the male mice expressing Xist is similar to the female wild type which is also expressing Xist. So again, this is in support of their hypothesis. And so not only do their pathology scores or severity match up, but the types of antibodies that they're producing are also similar. And figure 4 is looking at TAC-seq comparisons again. So here we're looking at the spleen CD45 plus cells or blood cells from these induced mice, this pristine treated mice in the immune deficient strain. So they saw a pretty good clustering of different immune cell types, or at least the sort of chromatin states that are similar to these cell types. And they saw that the fraction of these cell types, to me, they don't really look like they differ that much, but yeah, what are we supposed to take away from this? Okay, so this is just a baseline. Like we're seeing, okay, all the groups have these immune cell clusterings, and then we also see the frequencies. And then here we're looking at specific groups. So here it's a little hard to pull out, but I think you can pretty clearly see these green spots, which is the mutant male mice that had the high severity. So remember when I mentioned that they chose a pathology score of 10 to measure severity? They split that male group into two. They did the mutant male with high severity and then the mutant males with low severity. And I think it's a little hard to make the claim based off of this figure that these mutant male with high severity are similar to the wild type female. It's hard to see that green is overlapping with red. But I guess they're saying that this is happening. Again, as a control, they saw that, I'm not showing it here, but they have a figure showing that the mutant males and the females both had these Xist peaks. Also, again, I think it's pretty hard to read UMAP things besides trying to get like sort of groupings. I find it difficult, but the authors are saying that the wild type female and mutant male with high severity overlap in a B cell cluster associated with atypical B cell markers. Okay, so I guess they're going to focus more on atypical B cells, but let's see what happens. Okay, so figure 4, basically looking at a TAC-seq again, but with these immune compromised mice. And then figure 5, we're looking at RNA-seq. So here they did a similar sort of UMAP with the sequencing data. And again, they got really good clustering of different immune cells. And here, again, I think it's pretty difficult to see things that are overlapping. And so in here, this was in their supplemental, I added, you can see that in wild type female and the mutant males with high severity have this region that they're saying is atypical B cell sort of expression. They definitely overlap, right? Both of these are overlapping, but that's different from the wild type males and these mutant males with low severity. So that is, I think, clearer, which is pretty interesting, right? If you're seeing these expression, this section with similar expression levels, and they're related to the atypical B cells, that's a pretty interesting observation or result. And then here, they looked at the actual gene types. Yeah, so they looked at the upregulated genes and downregulated genes of, yeah, so they're comparing, so they're saying that these 19, I think that it includes all of them, not sure, but they're looking at the overexpression of these mutant males with high severity compared to wild type males. And they're pairing that with the overexpression of wild type females to wild type males. And they see this 35 genes overlap with overexpression of both the mutant males with high severity and the wild type females as compared to wild type males. And then they did that for the downregulation. They saw more genes in the downregulation. And so ZEB2 and CR2 are examples of, I guess, I'm not sure what the CR2 is showing, but here for the ZEB2, you can clearly see that this mutant male with high severity is similar to the wild type female. And with the CXCR5, you see the opposite trend. So you see very low expression in the wild type or in the mutant males with high severity compared to wild type females. And then, okay, so they're saying that they pulled out genes that are specific to atypical B cells, and then they did a similar thing with T cells. So they tested a bunch of different genes, but these are more representative of the overall. So in the T cell genes, you can see, again, there's more overlap in the downregulation. And yeah, I think you see the trend of these mutant males with high severity is similar to the wild type female expression. So again, I think ZEB2 and this CXCR5 are a very striking result. Like, you can see it, right? In some of these other ones, I think it's a little difficult to see the trend, but it's definitely there for some cases. So this is a sort of summary. I will probably go through my slides and edit this kind of stuff to make it a little bit better, at least have it in the same font. Okay, so the authors, based off of figure 4 and 5, which is the TAC-seq and the RNA-seq, they're saying that the mutant males with high severity is similar to wild type females in terms of B and T cells. So that's pretty cool, again, supporting their claim. And then figure 6, this is like more concrete evidence of the relation to human disease. So they got a bunch of sort of volunteers to give up their blood. So they pulled out, I don't know why it's 28 here. They say 130 of Xist-associated proteins were selected, and then 52 for a control proteins that are, so 52 here are antigens that are directly associated with DM. I definitely wrote down what these things meant. Oh, dermatomyositis. Dermatomyositis. Oof. I can't say that well. I'm sorry. DM, and then scleroderma. I'm so sorry. That's SSC. I will refer to them as DM and SSE, and then obviously SLE. So these 52 antigens are known to be associated with DM, so it's kind of like a positive control. Like we should see some type of overlap between the Xist and the disease. So we can actually get into this. So the controls, okay, so yeah, this is the thing that we're actually testing for. We're looking at autoantigens, and then we're comparing those to between diseased individuals and non-diseased individuals. So these 16 people were volunteers that gave up their blood for it, and then the other ones were collected from a human protein atlas. And here they're showing certain antigens that are significantly different between all of the diseased individuals, so DM plus SLE plus SSC, and the general public. And here you see they separated into each of the disease categories, and then from this they found that 9 of the antigens overlapped between all diseases. And besides this TRIM33, which is one of those controls for DM, like we know clinically people with DM have this autoantigen TRIM33, but all of these other ones, the 8 other ones, are Xist RNP components. So that's pretty huge, and they pull out 4 specific ones to look at. Let me see. So basically the takeaway from here, from these sections of this figure, is that these 9 or 8 new autoantigens are clinically relevant and should be tested to identify people with diseases. So usually the autoantibodies are expressed before the onset of disease, and so catching them early is really important. And so they'll make the claim that these are clinically relevant and should be tested for. This figure 7, they're looking at okay, so they're comparing mice with SLE and autoimmune patients. So, and seeing if there's shared autoantibodies. And so, I think C and D are specifically for these are specifically looking at mice. And so, they're looking at Xist RNP autoantibodies and seeing how the levels of them compare between the positive female control and then the test group. And at 12 weeks, there is a significant difference. The test group does not live up to the female positive control, but at 16 weeks, there is no difference between the females and the test group, which might indicate that this Xist expression does influence autoimmunity, but it might have a delayed sort of response. So it might take longer for these male mice to develop these autoantibodies, but they are still more similar to the females than the other groups, at least at 16 weeks. What the hell? Okay, well, we'll move on. Here, again, I need to edit my slide. I just wanted to get this out of the way because it's stressing me out. So here's a bigger picture. They did pull out specific genes that overlapped between males and or between mice and humans. Let me see if I can figure out how many. No, they do not say how many. But there is definitely overlap between these antigens in autoimmune patients and the mice that they're looking at. And overall, they're making this claim that having the expression of Xist makes you more predisposed to autoimmunity and can affect the severity of the disease. And yeah, so what I was saying before, these autoantigens that are part of this Xist RNP complex or whatever should be tested for in patients for autoimmune disease. So this is something that's like really clinically relevant, and they make the case that it's hard to sort of know this knowledge that like could help a lot of people, and to be like restrained in that. Like it's kind of unethical when we know that this exists and this would be helpful to not use it. So I expect some type of clinical outcome from this paper. I should say that this paper was published in Cell this year, February, and so it's very new. It's also, here, I'll just go here. So yeah, they're making a claim that we should, instead of, like for a lot of autoimmune patients, they do B-cell depletion, and that can obviously have pretty serious side effects, but they're saying here that it might be more useful to look at atypical B cells and to test for these new autoantigens.